It's been tradition for many years that we do end the session with the Pioneer Lecture, the Ann Shiras Pioneer Lecture. I'm going to tell you just a couple of things, again, about Ann Shiras as a matter of introduction. But once again, and this is to save me a lot of work reading, which you don't want to hear, Danielle and Jamie have gone to a lot of trouble to put up complete bios on the website for a number of the people who have contributed to BCA over the years. So those are detailed, and if you want to know anything about some of the key people starting from the very beginning, it's on the website, beautifully done. So I direct you to the website. But Ann Shiras was a pioneer medical photographer and one of the pioneers in BCA, and held many offices in BCA, and was definitely a part of the movers and the shakers to get BCA off the ground in the early days. Besides being a photographer, medical photographer, in the Pittsburgh area where she spent all of her life, through a whole 91 years. I hope to get to that. Several of you have already done that, plus. But she spent her time in and around the Pittsburgh area all of her life. She supported knowledge sharing, which is really an intricate key part of what BCA is about. She supported that not only with her actions, but also with some anonymous contributions in funds to help fund this lecture series. She also had donated some Polaroid stock in her later days to help fund BCA and push and promote the education and presentations like we're going to hear today. So it's in honor of Ann Shiras that the Pioneer Lecture is the final lecture, and I believe, as Chip told me to say, saving the very best to last. That's what you said, wasn't it? Something like that. Yeah, just following orders. But I really direct you to the website, the beautiful website that's being updated all the time, but the biographies of a number of notable BCA members are there. Now, it is indeed my pleasure to introduce the next speaker, Chip Hedgecock, which we all have seen his demonstrations and heard his talks, not only in this meeting, but in the past. He is not one of the 91 plus, but he's getting there. He's been a pioneer not only in his area of photography, of biomedical photography, as well as biology photography. He's been published, he teaches, that's his life. His life is teaching through images and teaching through workshops and other things. I had the pleasure of meeting Chip many years ago on my very first visit to Brooks Institute. And unfortunately, he drew the short straw to have to show this crazy kid from Texas around the Brooks campus, and that's when we first met. Which, yeah, it was a long time ago. I think we were both underage at that point. But anyway, ever since then, I've just been an admirer, certainly, of his work in natural history, natural science. I'm sure you have through publications in the journal and books and other things that he's done. So it gives me great pleasure to introduce the Ann Shiras Pioneer Lecture for 2025, Chip Hedgecock.
I want to thank you again, Gail, for that introduction. It's true, we did meet for the first time. I didn't draw the short straw. It was just something I really enjoyed, was sharing the Brooks campus with people when I was working there. I worked in the first year students black and white lab. And yeah, I met Gail, and it was striking to me because he talked about something I never heard before, which was a point light source and an enlarger. And I thought, well, in portraiture and stuff, we use cold light heads with a very diffuse light source, and he says, you should try a point light source, and I will show you grain in a colorless negative. And my brain is still exploded from that comment. So thanks. And it's a bit of a curiosity to me that given all the wonderful photographers in the room and in our association, and many of whom are truly pioneers, that I drew the short straw for this, but hopefully I'll make it worth your while. I had more to say, but now I've forgotten that. So I grew up here in Tucson. My family moved here from the Upper Peninsula of Michigan in 1959 when I was just a kid, about 2 years old. So I like to say I wasn't born here, but I got here as soon as I could. So these are my siblings. There are 5 of us. This is my oldest brother, David, my oldest sister, Robin, my annoying little brother, Kevin, my sister, Karen, and I'm the handsome fellow in the cowboy hat down here in the front. Still a fashion plate even to this day. So I grew up here, and I was introduced to the darkroom and photography in high school, as probably many of you were. And later went on to take photography classes at the local junior college, Pima Community College. I was a very bad student, so I couldn't get into the University of Arizona at the time. So I went to Pima College, studied photography there, and the teacher at the time was talking to somebody, and I overheard him say, if you want to study photography in Arizona, the best place to go is Arizona State University in Tempe. And so I thought, well, that's where I'm going to go. So after about 3 years at Pima College, I went up to Tempe, just outside of Phoenix, and studied photography at ASU for a while. And I liked it there. Art departments are interesting places. Students are interesting. But the big thing for me was that I became acquainted with some historical photographers that I feel really influenced my work a great deal. First being Edward Weston, who showed us first how not to do a mustache, but also showed us how with simple lighting and camera work you could make really elegant sculptural images of simple objects. And I've always found these very fascinating from the first time I ever saw them. Simple, everyday objects. Including probably his most famous image, Pepper number 30, who most people would recognize even though they maybe don't know who the photographer was. And the second photographer was Carl Blossfeld, who also showed us how not to do a mustache, but he had a nice hat, so that was cool. And he too, with camera work and lighting, made these beautiful, sculptural, iconic images that I was really taken with. So those 2 photographers really had a big influence on me, and still do to this day. So I was studying at the Arizona State University, and I began to decide, I'm really not learning in the art department the kinds of things that I feel like I really need to know in order to become a professional photographer. It was cool and all, but I wasn't learning. I wanted to learn studio techniques, I wanted to learn technical stuff. And so I'd always heard about Brooks Institute when I was in high school, and it was just a cool place that we talked about and dreamed about. And they're probably one of 3 really preeminent schools of photography in the United States in its day. So I sent in my paycheck, or my tuition check, and moved to Santa Barbara and started as a student there in 1979. So naive. So at Brooks, I got introduced to the 4x5 studio view camera, because pretty much every assignment we shot while in school was shot on a 4x5 studio camera, pretty much regardless of what it was. So I learned a lot about industrial scientific, portraiture, photojournalism. Photojournalism was probably the one exception. There was no Jimmy Olsen kind of stuff going on, so it was all shot on 35. But otherwise, everything was 4x5 view camera. And we would get an assignment like we have a metal object, you need to shoot it 3 different ways. And I was on the search and rescue team, and I had friends who ran a mountaineering store, and I would go there and borrow stuff from their store as props. So I was using things I was interested in, the outdoors, and that kind of thing, as props for a lot of my studio photography. So I graduated from Brooks with a degree in commercial photography, and I worked for a year running the black and white lab for first year students, and then I loaded up the truck, and I moved back to Tucson to seek my fortune, and spent quite a bit of time looking for a job. So this predates the internet, so all the jobs were promoted in a newspaper. So any job that even sounded like it might have photography involved, I went and applied to, all of which turned me down. Until about 1984, when I was hired by Gene McDermott, who some of you may remember, and Jim Foss was working there at the time as well, the photographer for biomedical communications at the University Hospital in Tucson. So this was never a job that I expected I might have. It never occurred to me, medical photography, but it turned out to be a really wonderful place for me. I learned a lot about science and photography, which would play a big role in my life from then on. And it allowed me to sort of use a lot of my studio techniques and things that I learned in school, photographing in the studio and medical kinds of things. So while I was there, I was also encouraged by Jim Foss and Gene McDermott to pursue my RBP. So RBP, probably most people know what that is. It's Registered Biological Photographer. It was a certification process that the FBCA at the time offered to photographers, certification, and it primarily, it felt to me at least, it primarily focused on medical photography, but I was able to sort of twist some of the assignments and use natural science as my subjects. And so this is shed skin from a Scolophus magister is the name, desert spiny lizard, which you've probably seen running around the grounds here, very large, very spiny looking lizards, probably doing push-ups for you. Had to photograph a bone to show texture, so instead of using a human skull or a femur, I used a deer skull. And I can't remember what assignment this was, but this is Manduca sexta. It's a tobacco hornworm moth, ovipositing on a tobacco plant. And I didn't know it at the time, but this animal, this moth, was going to become a very important part of my life as well. In 1927, at about 25 years old, Ansel Adams took this photo, monolith, the face of Half Dome. And it was for him sort of a sea change moment. It was a time when he began to truly think about pre-visualization, not thinking, oh, that's a cool image, cool thing, I want to make a photograph. It was, this will look cool as a print if I do this. So thinking ahead from the subject to what it would look like as an actual photograph. So pre-visualization, along with the zone system, were two things that Ansel was famous for and things that played a huge role in his life. So when Ansel took this, he first took it with a number yellow filter to try and darken the sky. And he thought again, you know, I really want the sky to be dark. I want it very dramatic. And so he put a dark red filter on and made this image here, which is iconic and famous, and probably everybody recognizes it. So I have this notion that we all in our lives have monolith moments. Moments in our career or in our lives where things occur to us, things appear for us, things that we try, but they're sort of sea change moments in our lives. So, excuse me, not on the scale of Ansel's monolith, of course, but this, for me, was what I consider my monolith moment. I was coming out of the hospital. I worked a night crew, and I found this non-native Mediterranean gecko feeding on insects outside the hospital under the light. I caught it with Stan Willis, who worked on the night crew with me. And I took it home, I set it up in my darkroom, and I made a series of images with my keep it simple, stupid lighting system that I learned at Brooks and I still use today. There's one light with a fill card. And I processed the film, I made a print, and I looked at it, I thought, this is really cool. I should do more of this kind of thing. And so for me, this is sort of the start of my sort of fascination, maybe even obsession with photographing small animals, sort of in the style of Edward Rustin and Carl Blasfeld, on film, trying to get that sculptural sort of iconic look with my subject. So in 1984, I got poached away from the University Medical Center where I was working. It's sort of a sad state that as you work someplace, especially in a I mean, even though that was a fairly large department, but you work your way up in the ranks and you come to a point where if you want to raise further up, if you want to make more money, you wind up having to become an administrator. I'm pretty sure administration was not something I was going to be good at, and it definitely was not something I wanted to be good at. I wanted to be a photographer. I wasn't willing to trade the camera for tassel blowers and a sport coat. I wanted to stay behind the camera. So fortunately, these two guys, Ed Arbus, who helped me with that Manduca sexta hawk moth overpotted on tobacco image, and I met him while I was working on my RBP. He, along with Jim Foss, really helped me sort of brainstorm about ideas for things I could do for my certification. And John Hildebrand, who was the director of the then department, Division of Neurobiology, Arizona Research Laboratories. It's sort of an interesting fact. So John Hildebrand was a Boy Scout when he was a kid. I was a Boy Scout when he was a kid. He got the photography merit badge. I got the photography merit badge. But his tutor was Doc Edgerton, who happened to live in his neighborhood. So for those of you who don't know, you've seen these sort of photos of bullets going through apples and balloons and things. So Doc Edgerton was pretty much the inventor of high-speed flash photography. So John had the fortune of having him as a mentor. I can't even remember who mine was. So Division of Neurobiology worked with insects nervous systems. They worked on a variety of insects, and so for me, interested in photographing insects and small animals, it was a wonderful place to be. So I got to photograph various lab animals, various things. I was still out looking on my own, finding things. And once in a while I would come into the office and there'd be a jar in my mailbox with a note saying, "Oh, I found this. It was trapped in my bathtub. I couldn't bear to kill it. I thought you might want to take it and take a photograph of it." And I did. It was really terrific. So this is just a side note. People often ask me if I put my animals in the freezer or something like that, cool them down before I photograph them, and I don't do that, with the exception being American cockroach, which is very big, very fast, not at all cooperative. So this was photographed on a light table, so putting it in the refrigerator slowed it down so it would cooperate for a little while. And then here's Manduca sexta. Manduca sexta, tobacco hornworm, was the primary research animal in this lab. And this is just a high-speed photo illustration to show the variety of flowers that were available in the desert for this moth to feed on. This is probably a pretty continental animal. I think it's everywhere in the U.S. at least. I also started experimenting with ultraviolet photography. So I was always frustrated by photographs of scorpions fluorescing under UV light that were blue, and I knew from my time in the field and handling scorpions that they fluoresce green, and people who attended my little workshop saw that with their very own eyes. So now you know that scorpions are green when they fluoresce under UV. And so I cannibalized a UV lab lamp, took the blue UV passing filter off the front, and duct taped it with some cardboard on the head of my Norman 200B, and experimented with filtration stuff until I was able to recreate pretty closely what scorpions truly look like under ultraviolet light. So this is bark scorpion. It's bifurcated tail, so what happens is it's incomplete twinning. Excuse me. So this is not something that happens infrequently in scorpions. It's just that, as probably many of you know, when insects grow, they have to shed their outer skin, their exoskeleton. So often scorpions that have this incomplete twinning, this bifurcated tail, they get hung up on their cuticle and they can't get out of it, and so they wind up dying. So not a lot of scorpions survive to adulthood with this condition. But this one, beautiful animal, which actually was a female that had a litter, what you call a bunch of baby scorpions, had a litter of baby scorpions, none of which had twin tails, but it was still interesting. And I continued pursuing my black and white photography, shot on film in my studio, using lab animals, using animals I found in the wild. Still using that simple lighting technique, trying to make sculptural what I felt were sculptural iconic images in black and white. So one of the cool things in this place where we are for this meeting, I like to call it sort of the southern four corners. So it's southeastern Arizona, southwestern New Mexico, northeastern Sonora, Mexico, and northwestern Chihuahua, Mexico. And it's called the Sky Island regions. And it's this confluence of the Rocky Mountains coming down from the north, Sierra Madre Occidental coming up from Mexico from the south. And in this area, the mountain ranges kind of break up, and it's called Basin and Range, Sky Island region. And so you have these mountain ranges that shoot up out of the desert grasslands, and they might go from 2,000 feet at the base to up to 9,000 or 10,000 feet at the peak. And so with that, you have a diversity of life zones, and also a diversity of plant and animal life. So it's this confluence of deserts and mountain ranges that comes together to make this truly a biological hotspot in North America. And in 2009, the Sky Island Alliance, which is a local Tucson-based not-for-profit environmental organization, through these remote mountain ranges, we would camp, we would stay there for a week or so, and then every day we would be out basically looking for our organisms. So we'd have botanists, herpetologists, birders, basically any discipline that wanted to participate would come. In 2017, the program got taken over by Greater Good Charities, and it became called the Drayon Discovery Expeditions. It's a little simpler to say. And they continued that project to today, although now it's been changed again, so now it's called the World Discovery Expeditions. So they're going to other places besides in the U.S. But all this time I've been invited along to be the primary photographer. Doing mostly what I was doing, volunteered for, is doing voucher photographs of specimens found in the field. So in the old days, they would catch a lizard or a snake, they would kill it, they'd put it in a jar, pickle it, put it on a shelf in a museum, and we don't do that anymore, thank goodness. Now you would catch an animal, you would do voucher photographs that show the animal's characteristics so you know what species it is, and then you would release the animal back where it came from. So I would be out in the day, most of the day, with my good friend and field buddy Dale Turner, who's a herpetologist, and we would find snakes and lizards and frogs and things, and we would take these photographs in the hand. It would be the characteristics needed to show what species this indeed was. So it might need to show scale pattern on the top of the head, might need to show scale pattern around the mouth, how many scales wide stripes are on the body, that sort of thing. And a hand shot to show sort of relative size. And after we had that, after we were sure we had photos that illustrated that, then we would do a beauty shot, something that would show the animal in a more attractive way. But we always did these first because often, maybe most often, the animal would escape and get away before we'd have a chance to do that. So this is the kind of thing we would shoot for at a beauty shot at the end. So this is a green rat snake that was found late at night, and it was brought back to camp in usually when you catch a snake or lizard, you put it in a big cloth bag. Early in the morning then, we took it back to the point of capture. It was chilly out, the ground was cold, the snake was cold, so it wasn't moving very fast. Reptiles are ectotherms, so they get their body heat from outside things, from outside surfaces or from the sun. So we were able to set it up where it was captured and get this nice photograph of it in its environment to show the animal well and its environment. So all of these are photographs that were taken after the handheld shots to show sort of the beauty shots of the animals we found. This particular snake, the Pacific Coast parrot snake, was found trapped in a 55-gallon barrel that a rancher had sunk into a dry riverbed to have water for his cattle. Ranching in Sonora is a big deal. But it became a death trap basically because a lot of things were dead in there. It's a pretty disgusting mess, except for this snake that was still alive, swimming back and forth, struggling, unable to climb up out of the 55-gallon drum. So we fished the snake out, and it's arboreal. Typically it would be up 20, 30 feet in the air on a tree somewhere, So I put it in a branch of a both-thorn acacia tree. Dale held it up with the mountains and things in the background, and I made this photo to sort of illustrate its environment where it might be found. Rockhorn lizard, or Ditmar's horned lizard. I've talked about this at a previous meeting, and maybe you're fortunate enough to get to hear me. So this was a lizard that basically was missing from science for almost 100 years. It had been found during the original border survey when they were initially trying to figure out where is the U.S.-Mexico border, and they put up the big obelisk along the way. And 2 samples were found. They were pickled, as was the custom at the time, and labeled Sonora Mexico. So Sonora Mexico would be like labeling it, you know, Arizona.
Yeah.
It's a huge area, and so there's no real specific locality data for where it was caught. And then after that, once in a while they would be found, but really there was no information on them. Nobody really knew where they came from or really knew anything at all about them. One of the students in our Mexico group found these on their family ranch in Sonora, Mexico. We started then a series of expeditions to go down there to research these lizards. We would catch them, weigh them, measure them, sex them, and record all those data, and then take them back to exactly where they were caught and release them back into the wild. And this became the focus of 2 or 3 expeditions. So I would be out in the field with Dale all day. I would come back to camp at the end of toward evening, and often there would be bags and things of other animals that other people had found and brought back for me to photograph. And so Dale would help me, and we would sometimes work late into the night photographing all these specimens that had been brought back so that they could be taken back the next day from wherever they were caught and released. And this is my studio setup. This is from the other natural science imaging book that Michael Perez edited. I did a chapter photographing small animals in captivity. So you can see it's, and if you saw my workshop thing, this all looks very familiar to you, but it's my favorite Vivitar 283 flash, which does exactly what it's supposed to do. That is, when I push the shutter, it flashes. It doesn't talk to me, it doesn't ask questions, it doesn't try to second guess me. It just does what I tell it to. A PVC pipe frame that holds a white ripstop nylon diffuser that I made myself. A series of plastic aquaria that I have on a Lazy Susan so that I don't have to try and chase an animal around the table while I'm photographing it. I can just rotate the container and stay where I am and photographing it. And then fill cards. So it's, again, I keep it simple, stupid lighting system, 1 light above and behind with a fill card. And I would set this up wherever it's convenient. So this is the tool shed of a ranch we were working on. This is set up on the tailgate of a truck. It's a smaller version of the same system with a magnetic light stand, ring stand, I'm sorry, that holds a little Vivitar flash with a sensor on it. This is in the living room of a ranch house that they let us stay in. This is a small group. There was only about 5 of us, I think, at this time. It was at the end of about a 2-week trip down there. And this was photographing a phasmid, a stick insect, that I wanted to show. It's really wonderful antennae. Hopefully you can see that here. It has really beautiful and long antennae, and it's very stick-like, wonderful body. Again, 1 light for the fill card. 1 light above and behind this fill card. This is more often the case. I would just basically pull all my gear out of the back of my truck. It would be out in the middle of nowhere, and I would just set it up in a relatively flat spot, set up all my diffuser, my flash, my fill cards, and all that kind of thing. And this was for a story, an article from the Journal of Biocommunications, which I've had a few. In this case, I was photographing talus snails, which are snails that live in rock piles and things on dry land, basically. I had no idea there were so many snails that lived on land until I started doing these kinds of things. And so this is kind of an iconic image for the Journal of Biocommunications, because it's the first time that they showed a can of Tecate as being part of your field gear. So these are all things that were photographed in that my little studio setup. All, again, one light above and behind with a fill card. Whenever we get to a site, I would have to go out and find soil, rocks, sticks, all the props I need from the environment where the animals would be found. So I could set up a realistic, true-to-life sort of nature fake, as my friend likes to call it, setting to photograph animals in. So this is a Ditmar's horned lizard. I talked about how we would collect data on them. Each specimen, we didn't give them names, we gave them numbers. So this is not the 55th animal caught that day, it's the 55th animal whose data are recorded. So we would just write the number on their tummy, we would measure them, usually measure the snout vent length, which is so for delicate audiences, that's the vent. So you would measure this length here from the snout to the vent, and then you would measure from the vent to the end of the tail. So there's two measurements. Then you would weigh them, sex them. This, I believe, is a male. Looks like a male. And that number then, next time they shed their skin, that number would come off with the shed skin. But we knew who was who and whose data went where. And I would do a dorsal and ventral shot to show color patterns on every animal. So we had all those data plus photographic data that all connected to that number to show that individual. This is a little Smith's black-headed snake. It's about the length of a pencil and maybe half as big around. Very small little snake. Typically it's fossorial. It would like to be under things in the soil or under rocks. So I dampened the soil a little bit, and the soil was very shallow as well. So it didn't have much opportunity to bury itself, because that's its first inclination. When you put it in the aquaria, it's exposed. It doesn't like that. It's going to try and burrow underneath the hide. Probably the prettiest snake I've ever gotten a photograph. Not king snake. They're really beautiful animals. More snails. Cool animals to watch. Yeah, they're fascinating. And beetles. So a variety of larger insects sometimes would come in, and I would photograph those. And this really shows the studio lighting and how important that can be. If you do tabletop stuff and you photograph shiny things, you know you need to give those surfaces something to look at. So that's what we have here. This is a large diffuse light source up here. This is the fill card across here, and then it wraps around, and you can see this part of the abdomen. If you didn't have something for that to see, it would just be black there. So you need to give those shiny dark surfaces something to look at. And I like photographing things in the hand. It gives you a sense of scale, it gives you a sense of their vulnerability, and it gives you a sense of how sort of delicate they are, how precious they are, how vulnerable they are. Yeah. So even this sulfugid, which looks very ferocious, and if you are a small insect, it is very ferocious, but for you, it's just this kind of scary looking, cool thing that'll just sort of walk around on your hand. They're very wonderful. Lumen prints. So Daniela talked a little bit about lumen prints. I started doing lumen prints about 2014 when somehow I stumbled across the process on the internet. You know exactly how that is. You start over here, and before you know it, you're over there somewhere looking at something else. And also my partner Kathleen Velo, who was teaching at the local junior college, she was teaching photography there, and she had a student that was doing lumen prints, and she invited me to come in and meet him and see his work, and I was just blown away by the process and thought, I got to try some of this stuff. And so in 2014 I began doing lumen prints, and I've been pursuing them off and on ever since. They're really a cool way to show plants and help get people to stop and look at things. I started doing bouquets, which give sort of a better sense of place to show sort of the plant community, the things that are living there in that area. And lots of other things that pass light, but are not botanical. In 2018, Kate Velo and I were both artists in residence at Biosphere 2, which is right up the road. Some of you maybe have had an opportunity to go up there and see this. It's the largest contained facility of its kind anywhere in the world. It's just over 3 acres that was totally sealed, and it's at the beginning they actually had what were called biospherians who moved into there and lived growing their own food, and all of that. Basically, it was an attempt to show a concept for moving to another planet, setting up a space station or something, and being able to survive, create your own food, and for an extended period. So this was a trial run for that sort of thing. It's now run by the University of Arizona, and you can go up there, go on tour, and they'll show you around, see all the cool stuff. Lots and lots and lots of really cool plants from all over the world growing inside this thing. So my partner, Kathleen Bilo, she does these wonderful underwater photograms. So she did those in the many different water sources that were within the biosphere, and I worked on lumen prints of the huge plant diversity. So this is one of Kate's underwater photograms. She'll go out into water in the dark at night, and this is actually how we met and got together. I was her assistant, and she would go out there, wade into the water, hold the photographic paper, color photographic paper under the water, and I would, when she told me now, I would pop the flash, and that would illuminate the water, what's in the water, the color of the water. It was really, she created just, to me, they're just these amazing, amazing images. And she'd done this all over the Southwest. And I did lumen prints. So this is a small banana leaf, but that's on 3 16 by 20 sheets of paper, so this thing's almost 4 feet long. You saw this image before when Daniela was talking about lumen prints. They had a swamp in there, with a mangrove swamp. And they have an ocean, which is set up to sort of emulate the Sea of Cortez. And this is a sea grape that was growing along the shore there. In 2019, I was invited to participate in 6 and 6. So they had 6 artists, 6 scientists. They teamed us up in pairs. And each pair would then come up with a project to do something that had to do with the Sonoran Desert or the Sea of Cortez. My scientist was Mark Dimmitt, who used to be the head of botany at the Sonoran Desert Museum. And he still is still a famous, locally famous botanist who does all kinds of weird plants and things. So he was my biologist, my scientist, and he had this notion of working on diaphanous plants, which are plants that are sort of peculiar to arid environments. Not a lot, or maybe tiny leaves, very skeletal structures that you can actually see through. And so this is a skeleton milkweed plant that's, again, about 4 feet tall, with 3 16 by 20 sheets. And this is how I set up for lumen prints. That didn't make a frame. It was 4 feet tall, like Daniela uses with hinges and stuff. So it's just a 1/4 inch thick plate glass and a 4 foot long sheet of plywood. And then I put spacers around, if I can get this to work. So we had spacers along here to try and keep it from crushing the plant too much, and these big spring clamps to hold it all down together. And it became another triptych, which I then retouched together in Photoshop to make a giant lumen print that then I had printed on sheer silk. So it's very, very sheer. Because of that, the image, when it's printed, it goes through, and so you can see the image on both sides, and when it's hung up, you can see through the actual, we call them veils. So I did these 4 veils of diaphanous plants in the Sonoran Desert, and they hung from lucite rods, and they were free at the bottom, so they would move as people walked around between them. So just like plants, they would move. As the air handlers came on, they would move. And you could go on the other side of them, and you could see the rest of the exhibit. So you could look through the diaphanous plants to see other parts of the Sonoran Desert that the other scientist teams had worked on. So diaphanous art about diaphanous plants. So I mentioned earlier that Greater Good Charities is now funding the World Biological Survey or something like that. I can't remember what it is. So I got invited to go along as a photographer to try out photographing in this nature reserve in northern Vietnam, just south of the Japanese border, and it's just sort of a little ways east of Hanoi here. And then this is where the nature reserve is. If you know about How Long Bay, that's right out in here, which is one of the 7 wonders of the world. It's really a beautiful, amazing place. If you get a chance to go see it. So I went there with total of 4 Americans. I was one of 4, and the rest, everybody else was Vietnamese, biologists, scientists, teachers, professors, students, and we had to walk a few miles in. We carried a lot of stuff on our backs. I carried almost all my camera gear. Everything else got piled onto these little motorbikes, which somehow made it through all these various water crossings and up really horrible trails. And so everything else was on these motorbikes, including a big basket of chickens, which were our dinner every night. So every night there'd be one less chicken. And this is what the trails on land looked like. And I could imagine if during the Vietnam War, if you're a 20-year-old somebody from the Midwest, you know, you go there and you can't see more than 20 feet in any direction, and you know out there somewhere are people trying to kill you, it'd be a really disturbing, frightening, scary experience. And we camped next to Bin Hong River, which translates to peaceful perfume, and it was really just a lovely place to set up camp for about a week and stay there and perform the various biological surveys. The one downside is they have leeches there, land leeches, and they're maybe 2 inches long or something. They would creep around and they would look for you. You'd see them doing this, like they're looking for somebody to bite. And so every night when you get into your tent, you would have to inspect your boots, inspect your pants, and try and flick them off before you got in, so they wouldn't, because, well, they always got in. In the morning you'd wake up and there'd be a big splotch of blood somewhere from leeches in them and feeding you on it during the night. So they had set up sleeping platforms that some people had set their tents on. We set ours up on the ground, and we used part of this sleeping platform as a studio for photographing stuff. I couldn't bring everything with me, so I couldn't bring the aquaria, and I substituted this small light dome softbox for my PVC pipe frame. And we set this up, and we would photograph all the animals that we brought back on this setting. And because I didn't have an aquarium, there would be me photographing and then 3 other people standing around like this to catch anything that tried to jump or run away. So it took a team effort to get photographs of everything. There's really beautiful big geckos there. More cool snails. This is really unusual because it has that sublumentum that goes up and sort of wraps around the shell. So this animal, the shell is probably about the size of a silver dollar or something maybe. It's pretty big. Really cool animal. Very few snakes. It was kind of chilly. The weather wasn't perfect. We found a lot of frogs and toads, only 2 or 3 snakes, and only a couple or 3 different lizards. But lots of frogs and toads. These cool skinks. They lived under the rocks right along the river's edge. Very fast, very hard to catch. Tropical vine snakes. So we have vine snakes here, and they're very similar to this. Very viney looking, very long and slender with a big head, and they have this gaping mouth that if they get frightened by you, they'll rear up and they open this great big mouth. But they're harmless. They're not venomous or anything. They're just trying to look like a badass and scare you away. But they're really cool, beautiful, elegant little animals. An unusual frog to me in the sense that it looks like a toad because it has all that sort of warty body, but it is a frog. To me, this is my favorite frog. Very handsome, I thought, with the sort of reddish brown panels and the creamy pinstriping and stuff. Very athletic, very willing and ready and able to jump or get away from you at any moment. But just really, to me, they're very beautiful animals. So I just got back from Mexico, Ditmar's horned lizard survey, working with the Miami Zoo. They want to capture Ditmar's horned lizards to set up a breeding program, captive breeding program, as well as a display. They would be the only zoo in the world that displays these animals, and they would have this captive breeding program that they could hopefully rear animals both as sort of an insurance policy for the future, but also just to learn more about these lizards, because nobody knows anything about them. So I have photos of them. I probably have the most photos of this animal anywhere in the world. And in terms of people who know about that, probably those are the people who know everything there is to know about Ditmars horned lizards, because they've just been rediscovered and just been started to get research. And again, we did dorsal, ventral photographs of all those things. And I meant to tell you that in that earlier photograph where I had the ventral surface with a number on his tummy, that animal was alive. It's a curious thing with horned lizards. You can turn them over and rub their tummies a little bit, and they'll just sort of relax and lay there on your back for a few seconds and let you photograph them. Very cute. And this is Rancho Las Playitas, which is a ranch where we were studying, capturing horned lizards, and that's Ditmars Hill, as we call it, in the back right there. We're looking for lizards. So I was a kid who, in high school, teachers and parents were sure I was destined to be wearing a paper hat for the rest of my life. And somehow I fell backwards into a job as a medical photographer, and somehow continued on to this day. And when they ask people, so what would you change in your life? I think, well, I really like where I am. I wouldn't want to risk anything by changing what I've done. So I'm really happy to have been here, to have gotten here. And I'll do this kind of stuff as long as people will have me, and as long as there's interesting projects to pursue. And I want to take a moment to talk a little bit about community. So we sit here in this room, we look around. This is one of our communities, all of these people here. Another part of community is when you step outside and you look around, and it's everything else that's out there, the plants and animals. So we're part of a greater community. This is part of the community that I'm especially interested in right now, today. And I want to thank you very much for having me. I want to thank Biocommunications Association. The FE education grants, I've gotten a couple of those. They really help a lot with funding some of these expeditions I've been able to participate in. Greater Good Charities and their Global Discovery Expeditions, who have been carrying on these expeditions, and the Skyline Alliance, who started it all way back when. Thank you very much.